Okay, so sorry for the abrupt ending to the previous video. Let's continue our discussion of Western blotting. Okay, so we have put our samples and our protein ladder into the wells of our polyacrylamide gel. Okay, and now what we are doing is we are establishing an electrical potential difference between the two sides of the polyacrylamide gel. Okay, so we're going to make this electrode down here the anode, or the positive electrode, and this electrode down here the cathode. Okay, so I want to make sure everyone understands what it means to have an electrical potential difference between these two plates. Okay, so electrical potential is a concept from physics. It is a mathematical model to understand reality, basically, and this is how the mathematical model goes. You ascribe to every point in three-dimensional space a real number, and this real number can be either positive, negative, or zero. Okay, so every place in three-dimensional space gets one of these numbers, and this number is called electrical potential. So in principle, you can build a machine, and this little man can have this little machine, and basically he can go and stand in different places in three-dimensional space and ask what is the electrical potential there, and the machine can measure the electrical potential. Okay, so the little man can go and stand on this plate over here, which is going to be the cathode, and he can measure electrical potential there. Now, when you have a metal plate, basically what happens is that all of the points of the metal plate end up in electrical equilibrium with one another, so they all end up at the same electrical potential, basically. So all of the points on this plate will have the same they'll all have the same electrical potential, and all of the points on this plate will have the same electrical potential. Now, at the moment, we haven't attached a battery or anything, so both of these plates are likely to be neutral, basically. So what we're now going to do is put a battery in between them. Okay, so where shall I draw this? So I'm going to have to highlight it so that we can see it distinctly. So here is a wire, and what I'm going to do is I'm going to put a battery between the two, like so. Okay, so I'm connecting both of these plates up to the two terminals of a battery. So here's one wire in pink here. Okay, here's another wire also in pink here. Okay, now basically a battery will establish an electrical potential difference between the two plates. Okay, so it will make one side have a lower electrical potential than the other. Okay, now the smaller arm of the battery, so a battery is denoted with two lines, a small one and a large one, and the small one will always be the side which has the lower electrical potential, and the larger one will be the side with the higher electrical potential. So basically, this side of the battery is going to have a higher electrical potential than this side. Okay, so we'll say this is the electrical potential of the negative side, so you would call this the negative terminal of the battery, and this is the electrical potential of the positive side. So we'll denote the electrical potential of this negative side of the battery, E negative, and we'll denote the electrical potential of the positive terminal of the battery, E positive. Okay, and there will be some electrical potential difference between these two. Okay, and all that means is how much does the electrical potential change if you move from one plate to the other, i.e. what's the difference between them. So that's what's meant by an electrical potential difference, or a voltage. So we can say what is the voltage, and what the voltage means is if you move from the negative terminal of the battery to the positive terminal of the battery, how much does your electrical potential go up? So basically it means the electrical potential of the positive terminal subtract the old electrical potential of the negative. So if you had a little man standing on the negative terminal and he had this machine that measured electrical potential there, he would get this value. If he then moved to the positive terminal and again measured electrical potential there, he'd get another value, E positive, and basically how much does the number change? That is what is meant by the voltage of the battery, or the electrical potential difference between the two terminals of the battery. Now when you connect the positive terminal to this plate over here, which is going to be the anode, what will happen is that they will all become electrically in equilibrium with one another. So the electrical potential of all the points on the anode and all the points of this wire will become the same as the electrical potential of the positive terminal of the battery. So they're all going to get this same electrical potential here. The same will happen on the negative terminal side, so all the points of the wire and all the points of this plate over here will end up with the same electrical potential, which will be this one here. So basically, these two plates will now have the same electrical potential difference between them as the two terminals of the battery, basically. Which means that this one is going to become our positive electrode, our anode, and this one will become our negative electrode, our cathode. So now there is an electrical potential difference between these two plates. This plate has a much lower electrical potential than this plate over here. Okay, and we're actually going to have an electrical potential difference between these two plates of 150 volts. Okay, so the unit of electrical potential is the volt. Okay, and therefore the unit of the electrical potential difference, or the unit of voltage, is also volts. So, what this means then is that the electrical potential of the anode here will be 150 volts higher than the electrical potential of the cathode. Okay, so basically these proteins are covered in negatively charged dodecyl sulfate anions, okay? And when you establish this electrical potential difference between these two plates, what will happen is the proteins will move towards the positively charged plate down here, so they're going to migrate down the gel towards the anode down here. Okay, now we're in a position to talk about bromophenol blue, and I should just put one last line to explain what I was just about to say. Basically, the reason that these proteins move towards the anode is because they're covered in negative charge, and negative charge wants to get to where the electrical potential is higher, which is now over here near the anode. Okay, so that's why the negatively charged proteins, which are covered in these dodecyl sulfate anions, will migrate towards the anode. Okay, so what we can now discuss is why we added bromophenol blue. Basically, bromophenol blue will also migrate towards the anode, okay, and it will migrate far faster than any of the polypeptides. So basically, what will happen is, in all of these wells, bromophenol blue will move down, 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 and basically, this is to allow us to make sure that we do not leave the gel electrophoresis happening for too long. Because let's think about what will happen if we leave this setup on too long. Well, what will happen is all of the proteins in all of these samples will eventually get to the bottom. If you give it enough time, all of them will eventually make their way through the gel and get to the bottom, and then you haven't spaced them out. You have to stop the process before the proteins get to the bottom, i.e. when they're all at different levels. Okay, if you give them all enough time, they will obviously all get to the end. So you must make sure that they don't get to the end. They have to be spaced out along the height of the gel. Okay, so the bromophenol blue acts as a marker to make sure that you don't let it get to the state where the proteins are all at the bottom, because if the bromophenol blue hasn't got to the bottom yet, it migrates quicker than all of the proteins, so that allows you to infer that none of the proteins have got to the bottom yet. So the bromophenol blue will appear as a blue line, which you can use to tell how far, well, the maximum possible distance which your polypeptides could have moved by now. Okay, and that allows you to make sure that the experiment is not going horribly wrong. Okay, right. So what will now happen is we will allow the gel electrophoresis to occur, so we'll allow electrophoresis to go on, and usually at this sort of a voltage you'll have to run it for 1 hour. That's the general running time for a gel electrophoresis with this voltage. Okay, now what you're going to do is, by this time, after 1 hour, what will have happened is the bromophenol blue will nearly have got to the bottom, okay, and you will now disconnect the voltage. Okay, so you'll take your battery out so that there is no longer an electrical potential difference between these two sides of the gel. Okay, and now your polypeptides will stop moving because there's no reason for them to move anymore. Gravity isn't strong enough to allow them to move through the polyacrylamide gel. Okay, so they will then stop moving, and what you're now going to do is you need to visualize it, because at the moment, what would have happened? Let's talk about what would have happened, but the problem is we can't actually see any of it. So in the protein ladder, we have all of these proteins of certain sizes which have been, well, which are all colored basically. So we'll have a nice spectral ladder here. So we'll be able to see this ladder. So maybe this one will appear green, maybe this one will appear red, sorry, pink rather, maybe this one will appear blue. So we'll be able to see these on the gel. But the actual protein samples that we put in, sample 1, sample 2, sample 3, sample 4, we won't be able to see the proteins in those because they were just proteins that we got out of a cell, and cells don't just label their proteins helpfully up for us with coloured tags. So instead what we're going to have to do is a way of actually visualising our protein. Now, remember, we started off just with a cell lysate, which contained a huge number of proteins. But let's say we are interested in just one of these proteins. We're interested in, do these cells express protein A, let's say. So we want to know, do the cells express protein A? Okay, so we specifically want to visualize the bands corresponding to protein A. Now let's think about what will have happened in these 4 samples. So, for simplicity, we are imagining that the samples just have 3 proteins in, but of course they'll have far more than that. Protein A, protein B, and protein C here. Okay, we've said protein A was the longest, protein B was the shortest, and protein C was somewhere in the middle. So protein B will get the furthest in an hour. Because it was the smallest, it will migrate the furthest. Then, next up, there will be protein C, so that might be somewhere back here. And then finally, there will be protein A. Now, I'm putting these bands on, but we won't be able to see these. These are just proteins, they're not colored tagged. We won't be able to see anything, basically. All we will see is a clear gel, basically. So we need to do something so that we can actually see the bands corresponding to protein A, which is over here. Now let's think about what it will be for for sample 2, sample 3, and sample 4. Well, it will be exactly the same, except that the bands will be a little bit smaller, basically. There'll be less protein in each of the bands, but the bands will be at the same height, basically. And as we go down, of course, the amount of protein in each one will go down more and more, basically, where 4, remember, is the 1 in 10 dilution compared to lane 1. Okay, so it will have a 1/10 of the protein in each one of these bands that each of the bands has in lane 1. Okay, but the bands should be at the same height, okay? But as I say, we can't see them yet. Okay, now what we're going to do is we're going to remove the gel. Okay, that's a quite simple operation. You just take the whole thing out, okay? Usually it will be in a plastic container. You'll then have to break open the plastic with some sort of knife, and then you'll take the gel out. Now what we need to do is gel transfer. Okay, so we're not going to visualize protein A, which is of course back up here. We're not going to visualize these bands corresponding to protein A on the original gel. Instead, what we're going to do is transfer it onto a membrane, and then we're going to visualize it on the membrane. Okay, so the next step is what's known as electrophoretic transfer, or simply gel transfer. Okay, and basically, I'm going to explain the principle. In reality, what you have is a beautiful machine that does it all for you. But this is the principle of what you need to do. What you will basically do is you'll take your gel out, and now I'm going to draw this picture as though we're looking from the side. So basically, my gel is this rectangle, like so, which is extremely thin, a few millimeters thin. Okay, we're going to take it up, turn it over and put it laid down like so. And I'm now just going to denote it like so. So we're now going to look at it from the side. So this is its thickness, basically. This is a few millimeters, let's say 2 millimeters. It will probably be about more around 3 millimeters, I'd say. Okay. So this is our gel here. So this is how I'm going to draw it now, and we're going to be looking from the side. Okay, so what we're going to do is basically we are going to firstly get a metal plate. Okay? The idea of what we are trying to do is we're trying to move all the proteins that are within the gel from the gel onto onto our membrane, and I'll explain the different types of membrane that you can use in a moment. Okay? So, how are we going to do this? Well, we're going to get the proteins to move in exactly the same way as we got them to move in the gel electrophoresis. We're going to use electrical potential differences. Okay, so, let's say this is our gel here. Okay? What we're going to do is we're going to put a special membrane underneath it, and this is the membrane we want all the proteins to go onto. So let's label things up in specific colours. So, let's have the gel in bright green here. Okay, then underneath the gel in blue, let's have the membrane onto which we are going to transfer the proteins. Now, what sort of membranes can you use? Well, there are two major membranes that you can use. You can use nitrocellulose membranes. This is kind of the old one that used to be used. Nowadays, it's more common to use PVDF membranes. Okay, and PVDF stands for polyvinylidene fluoride. Okay, so PVDF is short for poly and then vinylidene. Okay, polyvinylidene, I think it must be, fluoride. So polyvinylidene fluoride. Now let me show you the structure of polyvinylidene. I struggle with pronouncing that. Polyvinylidene fluoride. It's a very simple polymer again. So basically this is the repeating structure that you have. It's again 2 carbons, and then you have 2 fluorines coming off it. 1 fluorine here, 1 fluorine here, and 2 hydrogens coming off here. Okay, and this repeats itself again and again and again and again and again. Okay, right. So underneath the gel you'll have your polyvinylidene fluoride. Okay, and then underneath the polyvinylidene fluoride membrane, and I should just add that this is a membrane, okay, which will be the same size as our gel basically. It will be the same sort of proportions. So it's a, it's a thin little membrane, and you'll want it to be around the same size as the gel, so that the gel sits nicely on top of it. Okay, and then underneath it, what you'll put is a metal plate, and this metal plate is going to be our anode. So this is going to be our positive electrode. Okay, so here's our anode. Okay, then what you'll do is on top of the gel, you'll then put a little piece of filter paper. Okay, and then on top of the filter paper, you'll then have another electrode, which will be the negative electrode, which will be the cathode. So this is the cathode. Okay, and then in between the cathode and the gel, you then have your filter paper. Okay, so I'll colour in the filter paper in yellow, and I'll colour in the cathode in orange. So here in yellow, this is the filter paper, and then the cathode is in orange. Okay, so again, what of course you'll have to do to actually get these metal plates to have an electrical potential difference between them, what you'll need is another battery. Okay, and as soon as you connect each of these metal plates up to the different terminals of the battery, remember the big one is the positive terminal and the small one is the negative terminal, you'll end up with an electrical potential difference between these 2 metal plates. Now what is this going to do? Well, all the proteins are on the gel, and now what are they going to do? Well, they're all negatively charged, remember, because they're covered in the dodecyl sulfate anions. So they're going to migrate towards the positive electrode, the anode down here, because they are attracted to areas of higher electrical potential. So they'll move off the gel onto our either nitrocellulose membrane or PVDF membrane. Okay, so the idea is that we will get an exact replica of our gel, but on the nitrocellulose or the PVDF membrane. Now what's special about these nitrocellulose or PVDF membranes? Well, basically they are absolutely fantastic at binding to proteins. That is their unique selling point basically. They bind to proteins. So the instant these proteins come from the gel onto the membrane, whether it's nitrocellulose or polyvinylidene fluoride, they will then bind to that membrane, and basically you'll get hopefully an exact replica of the patterns that you have on the gel on the membrane, basically. And this sort of a preparation like this is called a sandwich preparation. And basically you just set up a sandwich preparation like this in a little machine, and then you just close down the lid, and 7 minutes later it will have transferred all the proteins from the gel onto the membrane. Okay, right, so that's electrophoretic transfer. Now, the next thing is that we have to image protein A on our membrane. So we're going to take out our PVDF membrane. We'll discard the gel now. We'll get rid of that. We don't need that anymore. And what we're going to do is image protein A on our membrane. And the way we're going to do this is using antibodies that are against protein A. Okay, so let me get another piece of paper. So basically, the idea is that we are now going to use antibodies which bind specifically to protein A to visualize protein A. Now there is a problem with this. If we just chuck antibodies onto this PVDF or nitrocellulose membrane, so let's say here is our nitrocellulose membrane now, and now we're no longer viewing it from the side, instead we're viewing it as, you know, a piece of paper or whatever it is. Okay, and on this we'll have, we'll have our protein ladder here that we can see, and we'll still be able to see that. So those proteins, of course, in the protein ladder will have moved on as well. Okay, so I'll colour this in again. And we'll also have the invisible four wells, basically, of our sample. Okay, so we'll have protein A, protein B, and protein C all spaced out nicely on this membrane, but we just won't be able to see it. Okay, so let me just finish colouring in the different members of the protein ladder. There we go. And now I'll mark where we had protein A, protein B, and protein C, so that we've just got this picture to be working with in the back of our head. Okay, so remember we had 4 separate samples, and the amount of protein of each of the 3 types will get smaller as you go from sample 1 down to 4, because remember we're doing dilutions. Sample 2 is a half as concentrated as sample 1, sample 3 is a fifth as concentrated as sample 1, sample 4 is a tenth as concentrated as sample 1. Okay, then we'll have protein B, okay, which was shorter than protein A, so it will have moved further. And there we go. And then finally protein A, not protein A, protein B, which, what have I done? No, protein A was the longest one, so this is protein A over here. Protein C was the middle one, wasn't it? Let me get my paper back to remind myself of what I said. Yes. Protein A was the longest one, so it went the least far. Protein C was the middle one, so that's here. And then protein B was the shortest one, so it went the most far. Okay, there we go. Right, so that's our copy of the pattern onto our either nitrocellulose or polyvinylidene fluoride membrane. Okay, now, the problem is, if we send in the anti-protein A antibody here, which is also sometimes known as the primary antibody, the problem is that it will just bind to the nitrocellulose membrane or the polyvinylidene fluoride membrane non-specifically, because remember I told you this membrane binds to proteins. Antibodies are proteins, so the antibody will just bind all over the membrane basically, and we only want it to be able to bind to the membrane if it's binding to protein A. So basically what we want is, if this is the membrane, we want protein A to be bound to the membrane, and I should have drawn it denatured, of course, that's very, very bad. Let me cross this out. So here is the membrane, then we've got our denatured protein A, and now we've got our anti-protein A antibody here. Okay, and basically we only want the anti-protein A antibody to be able to bind to the membrane indirectly, i.e. by binding to protein A. And we don't just want it to be binding all over the place basically. So what we're now going to do is we're going to do a blocking step, okay, and we'll continue this discussion in the next video.